Good morning all. I am Dr. PJ Madam Rafi. Today we are going to discuss about general considerations, means general concepts regarding antibiotics. Okay. What are those? We will see in the following slides. So first concept will be bacterial cell wall. Okay. Normally bacterial cell wall is composed of N-acetyl glucosamine, N-acetyl muramic acid and pentapeptides. Among with the help of these three compounds, bacterial cell wall will be formed. Okay. So these compounds will get synthesized? First will be this UDP N-acetyl glucosamine will convert it to N-acetyl muramic acid. Okay. So with the help of enzyme, that is enol pyruvate transferase enzyme. Here you have to remember the names and enzymes and inhibiting drugs also. Okay. So that's why I am mentioning each one. First UDP will be formed. This UDP NAG, that means N-acetyl glucosamine, will be converted to N-acetyl muramic acid with the help of enzyme enol pyruvate transferase. Okay. This enzyme will be inhibited by phosphomycin. Okay. Next will be now UDP NAG and that N-acetyl glucosamine and N-acetyl muramic acid formed. Now we needed pentapeptide for the formation of cell wall synthesis. Okay, that pentapeptide is formed from alanine racemase and alanine ligase. Okay, now this bacterial cell wall, if you see the picture here, this is the bacterial cell wall. Okay, so it consists of N-acetyl glucosamine and N-acetyl muramic acid. It forms along like this one. Okay, so these peptides elongation means in the this thing elongation of this chain will be happen with the help of enzyme, that is transglycolase. Okay, that's why elongation of this chain will be done by, that means NAG, NAM, next NAG, next NAM, next NAG, next NAM. This elongation will be helpful with the help of enzyme, that is transglycolase. Okay, now this elongation chain peptides will form. Now we need this cross linking. This cross linking will be done by transpeptidase enzyme. Okay, elongation will be done by transglycosylase. Okay, that's a major difference. Elongation will be transglycosylase. Cross peptidation means cross linking. This cross linking will be with the help of enzyme, that is transpeptidase. If you see here, this blue link will be transpeptidase. Okay, this is the peptide glycan pentapeptide. Okay, so for elongation transglycosylase, for cross linking with the help of transpeptidase. That's how cell wall will be formed. Okay, now we will see the inhibiting drugs. For enol pyruvate transferase, enzyme will be phosphomycin. For alanine racemase, and that means pentapeptide formation drug inhibited by cycloserine. This that means elongation of chain, that is elongation of chain will be done by transglycolase inhibited by vancomycin. Cross linking will be done by transpeptidase, which is inhibited by beta lactam antibiotics. Those are penicillins, cephalosporins, monobactam, carbamazepines. We will discuss each drugs in the following classes. Okay, now in this thing we have to remember the names of compounds, that NAG, NAM and pentapeptide, and the inhibitory enzymes in this slide. Okay, next major difference, we have to know the major difference between antibiotics and antimicrobials, bactericidals and bacterostatics. Normally antibiotics are obtained from microorganisms. That means antibiotics are naturally obtaining. Okay, natural things. Antimicrobials are synthetic compounds. These are drugs synthesized in the laboratory. That's a major fundamental difference. Antibiotics are naturally obtaining agents from microorganisms. Antimicrobials are synthetic. They are synthesized in the laboratory. Okay. Next thing will be bactericidal. Okay, and difference between bacteriostatic. Bactericidal is drugs which kill or irreversibly damage the multiplying bacteria. That means bactericidal means they mainly acting on the cell wall. Okay, they will inhibit the cell wall formation. Thereby they will cause death of the bacteria. Okay, so bactericidal means cell wall inhibitors. Bacteriostatic means they inhibit the growth and replication of bacteria. That's a major fundamental difference. Okay, here bacteriostatic it won't kill, but in bactericidal it will kill by inhibiting cell wall. Okay, here it will inhibit the growth. Okay, by inhibiting protein synthesis or DNA synthesis, somewhere in the cellular mechanism, cellular development, it will inhibit the growth only. But bactericidal it will kill the bacteria by inhibiting the cell wall synthesis. Okay, based on the next will be classification based on the action. Okay, first will be inhibition on cell wall synthesis. Obviously Cell wall means bactericidal. Examples of these bactericidal drugs will be penicillins, cephalosporins, monobactam, and carbapenems. Next one will be alteration cell membrane integrity. Obviously, in cell membrane integrity means they will inhibit cell wall synthesis. These are also bactericidal. Examples will be polymyxin B azoles. Next one will be inhibition of ribosomal protein synthesis. Okay, these are mostly bacteriostatic, but they will have some drugs showing bactericidal activity also. That's why they will be both. These drugs will have bactericidal and bacteriostatic activity. If these riboproteins means 50, if they are inhibiting 50s inhibitors, examples will be macrolides. It is a static drug. Clindamycin also static drug. Linezolid is also static drug. If it is inhibiting 30s ribosome, it is aminoglycoside means it is a cidal drug. Tetracycline is a static drug. Okay. Next is suppression on the DNA synthesis. Obviously DNA synthesis, obviously there will be no replication, so it will act as a bacteriostatic, but some drugs will show bactericidal activity. Okay, so what are those? Quinolones and metronidazole will be bactericidal activity. Nitrofurantoin will be bacteriostatic activity. Okay, here you have to remember the classification mainly. If they are acting on cell wall, what are the drugs? If they are acting on ribosomal, what are the drugs? If they are acting on DNA synthesis, what are the drugs? We have to see the names and remember the names. Okay, next concept will be drug resistance. How normally drugs will get resistance? First we have to know the what are the meaning of drug resistance. Drug resistance means unresponsiveness of microorganism to an antimicrobial agent after its repeated use. Okay, that means if you are using antibiotic repeatedly on a microorganism, it will become unresponsiveness. That means it is causing resistance. Okay, this resistance may be two types: single step mutation or multi-step mutation. Drugs which are showing single step mutation will be streptomycin and rifampicin. Okay. Next multi-step mutation will be erythromycin, tetracycline and chloramphenicol. Okay. Once drug resistance means drug resistant gene is formed, that resistance gene is transferred from one microorganism to other with three mechanisms, that is conjugation, or whether it is transduction or transformation. Conjugation nothing but attachment of one bacteria to another bacteria. It will be transfer the resistance gene. Transduction is that resistance gene is transferred from one bacteria to another bacteria. Transformation means through environment. Resistance gene is transferred through the environment. Okay. Conjugation means physical contact. Okay. Transduction means once the resistance gene is formed, that resistance gene is transferred to another bacteria. Okay. Transformation means it's through environment. Okay. Next, how this resistance will happen? What are the mechanisms in the resistance? Okay. Here we have to remember the names. They are asking the MCQs based on the if they will use other mechanism and asking for except, or you can ask the which mechanism. We have to remember the names. First is decrease affinity for the target. Okay, that means decrease affinity means once the normally these bacteria will have binding proteins. That means penicillin binding proteins like that they will have in the cell membrane. It will helpful for the binding of bacteria, this antibiotics to the bacterial cell wall. Okay, once it will binding to there, obviously it will enter into the cytoplasm, then it will cause death of the bacteria. Okay, that means killing of bacteria. So by decreasing affinity means decreasing binding capacity to that receptor. Obviously it will not bind means it will doesn't show any effect. What are the drugs? Okay, so what are the bacteria which will show resistance means MRSA. Okay, that is methicillin resistant Staphylococcus aureus. Then vancomycin drugs, they will develop resistance by decreasing affinity to the target. Means these bacteria will form alternate binding proteins. Okay, by developing alternate binding proteins, they will form resistance. They will get the resistance. Next one will be alternative metabolic pathway. Okay, by developing alternative metabolic pathway, bacteria will get resistance to which drugs? Sulfonamides. Next will be by developing some inactivating enzymes like beta lactamase penicillinase enzymes. These enzymes will degrade the drug, or whether it is antibiotic. So obviously when it is degrade the antibiotic, it doesn't show any effect on the bacteria. So there will be no effect. Obviously it will not show any response. Then there will be no killing of bacteria. So what are the drugs which will showing by inactivating enzymes means aminoglycosides, beta lactams and chloramphenicol. Next will be decrease drug permeability. Drug permeability means when drug is bind to the receptor, it has to get permeable into the intracellular. Okay. If the drug is not going to intracellular, obviously there will be no response, right? When it is binding outer surface, it has to go inner surface, then only it will show some response. So when some bacteria, they will not permeable, they will form resistance. Okay, what are those drugs? Aminoglycosides and tetracyclines. Last one will be efflux pumps. Efflux pumps means they are present in the bacterial cell wall. They will efflux the drug out of the body. Okay, so when they efflux some blood from the cellular to the circulation, it will get excreted. Okay, why means these are the pumps, they are not allowing, or they are not allowing to the antibiotic to the intracellular. So they are effluxing intracellular antibiotic to the circulation. Obviously, when out of the, it will come out of the bacteria, it doesn't show any response or any action on the bacteria. So obviously it will form resistance. What are the drugs means? Tetracyclines, erythromycin and fluoroquinolones. Okay, next. Last thing, it is very important. Principles of antibiotic dosing. Okay, these names will repeatedly asking in MCQ wise. Okay, that is minimum inhibitory concentration, concentration dependent killing, time dependent killing, post antibiotic effect. We have to remember these names and what are those meanings also. Okay, first of all, minimum inhibitory concentration. Minimum inhibitory concentration means it is the lowest antibiotic concentration that prevents the growth of microorganisms even after 24 hours incubation period. Okay, if you see the name, it is the lowest, it is the least one, least antibiotic concentration that prevents the growth of obviously microorganisms. Okay, that is called minimum inhibitory concentration. That means it is the least concentration where it will inhibit the growth, that is minimum. Next is concentration dependent killing. Concentration means we have to give the drug in high concentration, then it will kill. Okay, that means killing effect of the drug is high when the minimum inhibitory concentration is more, that means high level. When minimum inhibitory concentration is high level, the drug will have high killing effect. Okay, that when how we will get the minimum inhibitory concentration high? We have to give the drug in high doses in single large dose. Okay, what are the drugs which are showing concentration dependent killing? Aminoglycosides and fluoroquinolones. Nothing but simple. If you are giving a drug in one dose only one day, that means you have to give the drug dose in very high quantity. Okay, that's why when you are giving in high concentration, it will show high response and it will kill the drug, it will kill the, sorry, it will kill the bacteria easily. So those drugs will be aminoglycosides and fluoroquinolones. We have to give these drugs in high concentration. That means we have to give in once a day. Okay. Next one is time dependent killing. Time dependent killing is antimicrobial actions of drug depends on the length of time. Okay. The concentration remains above the minimum inhibitory concentration. That means here also the minimum inhibitory, the drug concentration should be above the minimum inhibitory concentration, but it should be sustained prolonged time. It should be there in a longer time. If you see in concentration dependent killing, where we have to give the drug dose in high quantity. Here we don't need to give high quantity. We have to maintain the concentration high above the MIC for such a long period, like 24 hours, like that. Okay, so we need to give drugs, doses will be in different times. Okay, that's why we have to give drug in multiple doses. Okay, so by giving multiple doses, obviously what will happen? When it is decreasing, we are injecting the drug, so obviously it will be maintaining above the MIC for such a long period. Examples will be beta lactam and vancomycin. Okay, that's the fundamental difference. Okay, in CDK and TDK, that concentration dependent and time dependent, in this both, that drug concentration should be above the MIC. That's the fundamental difference between these two and post antibiotic effect. Okay, here the minimum in the drug concentration should be above the minimum inhibitory concentration. Okay, in concentration dependent killing, we will give high dose at a once. In time dependent killing, we will give doses in different, that is multiple doses in a day. Okay, next is post antibiotic effect. Post antibiotic effect is the inhibitory effect of antibiotic is present even after when their concentration is below MIC. This is a fundamental difference. Okay, here the antibiotic effect will be sustained even it will be below MIC. Okay, below MIC also it will prevent the growth of bacteria. That's the fundamental difference between post antibiotic effect and CDK and TDK. Okay, examples of drugs showing post antibiotic effect is penicillins, cephalosporins, carbamazepines, vancomycin, tetracyclines, aminoglycosides, linezolide, rifampicin, sulfonamides, and fluoroquinolones. These are the examples. You have to remember that these names MIC, CDK, TDK, post-antibiotic effect, and the drugs which are showing CDK and TDK and post-antibiotic effect. Okay, that's all. Thank you.